Dear students, today I am going to discuss about the topics generation of T cell receptor diversity by genomic rearrangement and then structure of B and T receptors. So first I will start with the T cell receptors. Here you will see that the adaptive immune system, the acquired immune system combat pathogens with the help of very specialized antigen receptors. Adaptive immune system can provide long-term immunity against a particular pathogen and is very specific in its response. Therefore, it is not surprising that its antigen receptors are also highly specific in their interaction with the antigenic determinants of the pathogens. And these antigen receptors are called either the B cell receptors or the T cell receptors, depending on whether they are found on the surface of B cells or T cells respectively. So the T cell receptor is a protein complex found on the surface of T lymphocytes that is responsible for recognizing fragments of antigen as peptides bound to major histocompatibility molecule. So initial description of TCRs were made 30 years ago, primarily through similarity with immunoglobulin DNA sequences. From this beginning, a clearer picture of TCRs as a pair of clone-specific heterodimeric polypeptide chains consisting of both constant and variable region here has developed. In humans, the majority of T cells express TCR composed of alpha and beta chains, and a smaller subset of T cell express TCR with gamma and delta chains. The TCR, though, through its complementarity determining regions, which is known as CDRs, endow the T cell with the ability to recognize and respond to the foreign material, which is known as antigen. And first, the antigen presenting cells digest the pathogen and display their fragments on major histocompatibility complex molecules. And this MHC antigen complex binds to the TCR while other co-stimulatory molecules like CD28 are activated, leading to T cell activation, then proliferation, differentiation, apoptosis, or cytokine release. So the MHC antigen complexes are, however, not the only molecules capable of interaction with the TCRs. Non-peptide antigens such as lipids can also interact with TCRs via some of the 5 isoforms of CD1, that is A to E of the isoform of CD1. And several studies describes that the TCRs binding to metabolic intermediates bound to the MHC-like molecules MR1. So TCRs have been described as the most integrated receptor structures of the mammalian immune system. And now here we are going to discuss about how these unique structures are developed, their mechanisms used to drive immune response via TCR signaling, and the effect of their effect here. So, like all the all other receptors, the antigenic receptors also have one domain that recognizes its ligand, or that may be in the case of antigen. and another domain which transduces the signal into the interior of the cell. So this is the function of the receptor. As would be expected, the antigen recognition domain differ greatly, while the signal transduction domain shows very little variation between different antigen receptors. So now I'm coming to the types of TCRs. There are two types of TCRs. One is TCR alpha beta and another one is TCR gamma delta. So TCR alpha beta receptors, they possess two polypeptide chains. One is alpha and second one is a beta. And gamma delta also contains two polypeptide chains. One is gamma, another one is delta. The T cell bearing TCR alpha beta receptors are much more numerous than those bearing the TCR gamma delta receptor. And the selection of antigen specificity of TCR alpha beta receptor is also much higher compared to that of the TCR gamma delta receptor. So it is noteworthy here that the innate immune system also employs receptors to recognize pathogens. And these receptors are known as pattern recognition receptors. The most widely studied pattern recognition receptor is a toll-like receptor. Now I'm going to discuss about the differences between the pattern recognition receptor and TCRs and BCRs. So as I said, the pattern recognition receptors belongs to the innate immune system, and BCRs and TCRs belongs to the adaptive immune system. PRRs show limited variation in their pathogen specificities and have a very few subtypes, but on the other hand, BCRs and TCRs show extensive variation in their antigen specificities. They recognize pathogen-associated molecular patterns, which is known as PMPs, which are common molecules present on the surface of most pathogens. On the other hand, BCRs and TCRs recognize specific antigens on the surface of particular pathogens. Since PPRs recognize common molecules on the surface of the pathogen, they are not pathogen specific. They're very much non-specific. But here, both the BCRs and TCRs are very specific. They are antigen specific and can even distinguish between closely related pathogens. PPRs expression do not involve DNA rearrangement, but here BCRs and TCR expression involve DNA rearrangement between genes at the immune receptor loci. So now how I'm coming to the TCR in detail. So TCRs, as already discussed, these receptors are present on the surface of the T cells. The initial experiments involving characterization and gene identification of TCRs were much more difficult than those for BCRs. And this was due to a variety of factors, all of which delineate the differences between BCRs and TCRs. And the main hurdle faced by the immunologists while performing pioneering studies research on TCRs where the biochemical assays involved in characterization of any macromolecules are very easy to conduct if it is found in the soluble form. Since the TCRs doesn't have a soluble variant, the biochemical assays is very difficult, really very difficult to perform. A fact which was one of the main reasons for the delay in the characterization of TCR. Next one is the antibodies, and the BCRs can recognize the antigen directly. The simple antigen binding site assays could be used to isolate or purify the receptor and for advanced characterization studies. But TCR, on the other hand, recognizes the antigen only when it is bound to the MHC molecule. So designing assays for such a system is extremely challenging, and therefore makes the isolation and purification of TCRs more difficult. Then plasma cells expressing antibody molecules transcribe immunoglobulin genes at a high rate and produce many copies of mRNA encoding immunoglobulin chains. This facilitated the identification of immunoglobulin genes. On the other hand, the transcription of TCR genes in T cells occur at a very low rate and produce few copies of TCR mRNA. Thus, only a small fraction of the total mRNA of the T cell codes for TCR, and this made the identification of TCR genes a very difficult attempt. Now, I'm coming to the TCR development and diversity. So the TCR is disulfide linked membrane bound heterodimeric protein, normally consisting of the highly variable alpha and beta chains expressed as a part of a complex with the variant that is the CD3 chain molecules. And this CD3 is not at all a variant, it's an invariant molecule. So the variable part is the alpha and beta chains. T cells expressing these two chains are referred to as the alpha and beta T cells, though a minority of T cells express alternative receptors formed by the variable gamma and sigma chains, so referred to as gamma sigma T cells. So TCR development occurs through a lymphocyte-specific process of gene recombination that assembles a final sequence from a large number of protein segments. And this genetic recombination of TCR gene segments in somatic T cell occurs during the early stages of development in the thymus. TCR alpha gene locus contains the variable and the joining gene segments as VV and JB, V beta and J beta, whereas the TCR beta locus contains a B gene segment in addition to V alpha and J alpha segments. Accordingly, the alpha chain is generated from VJ recombination, and the beta chain is involved this VDJ recombination, and here we have seen the VJ recombination. So this is similar to the development of gamma delta TCRs, in which the TCR gamma chain is involved in the VJ recombination, and the TCR delta gene is generated from the VDJ recombination. So recombination is temporarily regulated by the access of some protein, some gene, that is the recombinant activating genes, the RAG gene. There are two types of recombinant activating genes. One is the RAG1 and RAG2 to recognize sequences. TCR recombination occurs at two stages during the process of T cell development. First, the beta chain gene undergoes V beta and J beta rearrangement before V beta and DJ beta recombination in the double negative cells of the thymus, and rearrangement of the alpha chain takes place in double positive thymocytes. Then RAG1 and R2 bind to and introduce double strand breaks at recombination signal sequences, which flank all TCR gene segments, and the DNA repair machinery completes the recombination reaction. So RAG1 or 2 expressed, RAG1 or 2 expressed first bind to or expressed by all the lymphoid progenitors and immature T and B cells. The TCR alpha gene locus consists of 46 variable segments, 8 joining segments, and the constant region. Beta chains, TCR beta chain locus consists of 48 variable segments followed by 2 diversity segments. and 12 joining segment, and 2 constant regions. The TJ segments are located within a relatively short 50 kb region, while the variable genes are spread over a large region of 1.5 megabases. Therefore, the process of VDJ recombination requires contraction of the entire locus by looping. The ultimate goal of the recombination process is this diversity among the CDRs. And CDRs are the antigen binding section of TCRs, and uh, diverse recognition capability leads to efficient protection against pathogens and the generation of optimal immune responses. So here VDJ recombination along with the addition of or a deletion of nucleotide at the junctions between gene segments helps to generate uh, an, an astounding amount of TCR diversity. In addition to the diversity of TCRs, their cross-reactivity also contributes to host defense. And cross-reactivity of TCRs means giving a single T cell host to ability host which have the ability to react to multiple peptide ligands. And this is achieved by a degree of flexibility in TCR and peptide binding. This means fewer T cells are required to detect foreign materials. Additionally, antigen may be detected by several T cells, leading to polyclonal responses, which will make pathogen escape from more difficult. And this wide-ranging cross-reactivity is not without its problems as a concept of T cell being activated by antigen and then cross-reacting to self-ligand is possible. And this particular thing is known as molecular mimicry and is represent on theory of root cause of autoimmunity. So then we are coming to the characterization of TCR. So some of the earliest studies regarding the characterization of the TCR establishes the phenomenon of self-MHC restriction in TCR. In this regard, the studies by Doherty played a crucial role. This researcher provides that TCR of an animal recognizes the antigen when it will bound to the animal's own MHC molecule. So there comes the restriction. So this is known as self-restriction. So T cell can only be activated by an unique peptide associated with self-MHC. There are two models. One is known as the dual receptor model. Here, two receptors are there. One bind with the antigen, another bind with the MHC molecule. And the second one is the alternate self model. So one receptor present there that recognizes both the antigen and the MHC molecule. Next is isolation of TCR. So the earliest experiments regarding the isolation of the TCR protein were performed in the labs of Kaplan and Marek. And they have described that when they immunized a mouse with ova, here, mouse 1 with ova, that is ova albumin. And after a few days, isolated T cells from this mouse. These isolated T cells contain a subpopulation of T cells with over-specific tissue. These cells are short-lived, and therefore to increase their lifespan, they were fused with cells from the T cell tumor, and the hybrid cells formed after such fusion are known as T cell hybridomas. And these T cell hybridomas were diluted, and then and the clonal population of from each different hybridoma cells was grown separately. Then the clonal population of the which was specific for oval booming and hence got activated by the in response to OVA was injected into a new mouse here, mouse 2. After a few days, B cells were isolated from the spleen of the mouse 2, and some of these B cells generated antibody against a OVA-specific TCR. These B cells were then fused to cells from B cell tumor to form a B cell hybridoma. Out of these B cell hybridomas, the one that produced monoclonal antibody against OVA-specific TCR was identified. This monoclonal antibody was then used to precipitate out the OVA-specific TCR from the solution containing all the membrane proteins found on T cells. And this technique is used, this technique of using specific antibodies to precipitate out its antigen is known as immune precipitation. The importance of this experiment by Kaplan and Marak was that it allowed early immunologists to isolate a TCR specific for a particular antigen, is oval booming. And this seminal experiment facilitated further investigation and characterization of the species. Now I'm coming to the identification part of the TCR gene. So in 1990s, few researchers were published few of their data, research data, and from these landmark discoveries that revolutionized the field of immunology. One particular group, that is TACMAC group, discovered the gene sequence for the TCR beta chain in human, while Davis and Hedrick group discovered the same in mice. The strategy described by the latter research article was especially elegant. And the biggest hurdle is this study here was the low expression of mRNA encoding the TCR. So what David and Hedrick hypothesized that mRNAs encoding in TCR must be found on membrane-bound polysomes. So thus, by isolating only the membrane-bound polysomal mRNA from the T cell hybridoma, they enriched their sample for mRNA encoding TCR, and then they prepared phosphorus p32 level cDNA from the complementary DNA from the enriched mRNA. And the enrichment was an important step, as it reduced the number of cDNA clones to be screened at. Next, they isolated membrane-bound polysomal mRNA from the B cell hybridoma, and this was followed by hybridization of the radiolabeled cDNA from the T cell hybridoma and mRNA from the B cell hybridomas. For the genes expressed in both T cells and B cells, that cDNA and mRNA hybridize together to form double-stranded structures. On the other hand, the cDNA molecules which remained single-stranded were those which are expressed only in T cells. So these single-stranded cDNA molecules were cloned, and it was hypothesized that any cDNA clone encoding a TCR gene would show a changed DNA profile in T cells compared to the cells from other organs. Thus, certain hybridizations were performed with DNA from various cell types using these cDNA clones as probes. This led to the identification of a cDNA clone which showed different DNA bands in non-T cells and T cells. Further, when probed with a cDNA form from the clone, The T cells with different antigen specificities also show band profiles which are very different from each other. So these results pointed to the fact that the clone of cDNA is formed as a result of DNA rearrangements. Subsequent studies established that the clone we have got from cDNA clone encodes for the TCR beta genes. Next, we are going to discuss about the structure of the TCR complex. So all the TCR chains possesses a variable and a constant read domain. Another common feature of all the receptors is that they possess two key components. One of the components is responsible for ligand binding or ligand recognition, while the other is responsible for signal transduction across the plasma membrane. The variable regions of all the TCR chains together form the antigen recognition site of the TCR. So, on the surface of the T cells, the TCR is found in association with the CD3 complex, and and the resulting complex is referred to as the TCR CD3 complex. The CD3 complex is involved in signal transduction and is composed of 6 polypeptide chains, which are recognized as two zeta homodimer, and then gamma epsilon and delta epsilon heterodimers. Although they bear the same names, gamma and delta chains of the CD3 complex differ greatly from the gamma and delta chains of the TCR. So the polypeptide chains of the CD3 complex contain a long cytoplasmic tail, and this cytoplasmic tail helps in signal transduction. Next, we are coming to the B cell receptors. So B cell receptors consist of an antigen binding transmembrane immunoglobulin, that is MIG, in complex with two transmembrane polypeptide that is named as Ig alpha and Ig beta, containing the immunoreceptor tyrosine activating motif, which is known as ITAMs, and which enables the transmission of the intracellular signaling. So like the other immunoglobulin, they are the monomer. Here you can see they have both the chains, light chains, here the light chains and the heavy chains. They have the variable region and the constant region of both the light chain and heavy chain. So this is the variable part and this is the constant part. And the light chain consists of two polypeptide chains, lambda and kappa. And the heavy chain consists of 5 polypeptide chains, that is mu, lambda, epsilon, alpha, and gamma. So this is the structure of B cell receptor or B cell receptor, and here you can see this is a specific structure of transmembrane IgM that is functioning as a B cell receptor. Here you can see this transmembrane region. This is the membrane bound region of the protein or polypeptide chains, and this is the cytoplasmic tail that is functioning in signal transduction process. So all these antibodies, they are grouped into 5 different groups. So IgM, IgE, IgD, IgA, and IgG, they have different functions. Like IgM express effective killing in killing bacteria. IgE is responsible for the mediator of allergic responses like influenza, fever, asthma, hives, like that. So IgA found in saliva, in tears and milk. IgG found most abundantly found in serum. Now the BCR pathway. So the pathway means BCR activation starts when the antigen or the pathogen aggregate with the BCR. So when they aggregate with the BCR or the antigen binding site, then the motif, ITAM motif, they contact with that, and then they express release of a particular protein kinase. So this protein kinase will then activate the proteins of known as FYN, LYN, and BLK proteins. These proteins belongs to the SRC family protein. And then ITM tyrosyl residues phosphorylation took place by a particular kinase which is known as tyrosine kinase. Subsequently, SYK translocation from the cytoplasm to the membrane took place, which activates the RAS pathway. Here you can see different type of biochemical intermediates, different type of enzymes are functioning. This may be different pathways they are performing, and they are also expressing in so many transcription factors are expressing. So here mostly the SYK is a phosphorylated form of a complex with the phosphorylase. This is phosphorylase 3. So they are actually activating this and initiating the pathways. One of the pathways mentioned here is the RAS pathway. There may be other pathways, that is ERK, JNK pathway, PKC pathway, calcium dependent pathways are also there. So the last one is the difference between the TCR and BCR. So as we said that the TCRs are present at the surface of the T cell, BCR present at the surface of the B cell, and the antigen recognition component composed of either alpha, beta, or gamma, delta. Here, two heavy chains or two light chains, but both belongs to kappa and lambda chain. So signal transduction component in case of TCR is CD3, but in case of BCR it is IgAM or Ig alpha or Ig beta. So they are mostly they are non-soluble or insoluble. Here they are soluble as because they are mostly they are antibody. And they contain two identical antigen recognition sites in case of TCR, but PCR contain only one antigen recognition site. Here you can see that they encoded by different genes. Here they encoded by IgH, IgL, and IgK loci. Recognizes only by antigen itself, but they recognizes antigen when the antigen complex with MHC. And they do not undergo any somatic hypermutation, but the BCRs undergo somatic hypermutation, so that the BCR can change its amino acid sequences after it comes in contact with the antigen. That's all.